Hello everyone, welcome again to Julsummit Biochemistry Hub. Today we'll be learning about antibiotic resistance. Antibiotic resistance is the reason most of our antibiotics are not working. What's the problem? Every living thing has the ability to respond to the environment in a bid to survive. Everything that lives, including microorganisms, are evolving to be able to increase their rate of survival. While we take drugs to deal with these microorganisms causing diseases, which we call microbes, they also want to survive, need to survive, and also they are also developing strategies to avoid the drugs that we take, overcome the drugs that we take, which they see as threat to their survival. Antibiotic resistance happens when these microbes have evolved, upgraded. They've been able to develop strategies, acquire the ability to withstand the effects of these drugs that have been specifically designed to inhibit or destroy them. So when they can live comfortably in the presence of these threats, we say antibiotic resistance has taken place. Shortly, I will be showing us biochemical mechanisms which these microbes use to survive the effect of our drugs and continue to perpetuate their harm while we struggle for our own survival. Number one, biochemical mechanisms by which antibiotics resist, the microorganisms resist antibiotic drugs is inactivation of drugs by microbial enzymes. These microorganisms are able to destroy, inactivate, render the drugs we take against them inactive, ineffective, by producing enzymes that can degrade these drugs. An example is S. aureus or Staphylococcus aureus and Escherichia coli. These microorganisms, amongst others, can produce beta-lactamases. These are enzymes that can cleave the beta-lactam ring in drugs like penicillin and cephalosporin, thereby rendering these antibiotics ineffective and prolonging the period of illness. Another microorganism that can destroy antibiotics by their enzymes are the M. tuberculosis, bacteria that causes tuberculosis in humans. They have the ability to produce the enzyme acetyltransferase in the presence of anti-tuberculosis drugs like isoniazid, thereby rendering this drug ineffective. Number two biochemical mechanism of antibiotic resistance is the altering of drugs' targets. Microorganisms can alter the targets of these drugs. Take, for example, methicillin-resistant Staphylococcus aureus. The target of the drug of penicillins and most beta-lactamases is a protein. So these MRSA can modify, change the structure of these penicillin binding proteins so that beta-lactam antibiotics cannot bind. And without binding, it cannot function. And they don't function, there will be no therapeutic advantage. Also, M. tuberculosis, another bacteria, produces variant forms of RNA polymerase. Why? Rifampicin works against tuberculosis by binding to RNA polymerase of the bacteria. So the bacteria, in a bid to survive, changes the target of the drug so that these drugs can bind. And without binding, drugs do not function effectively. And lastly, also, as an example, under the altering of drugs target by microbes, are bacteria that resist fluoroquinolones. This drug, fluoroquinolone, binds to DNA gyrase of microbes. That is where they bind to function. And so these bacteria that can resist them must be able to change the DNA gyrase of their cells so that fluoroquinolones cannot bind. And when it cannot bind, it cannot bring therapeutic effect. The third biochemical mechanisms by which microbes resist antibiotics is by reducing the uptake of these drugs. Drugs need to enter microbial cells for them to function effectively. And bacteria like the Pseudomonas aeruginosa has been able to reduce the number of porins on its cell membrane. Why? Drugs as carbapenems, e.g. imipenem, enters the cells of these microorganisms through their porins. So, for them to survive these drugs, they reduce by themselves the number of these openings on the cell wall, thereby reducing the uptake of drugs like carbapenems, thereby rendering them ineffective. Also, another bacteria, the mycobacteria, changes its cell membrane to a form that is rich in lipids, so that the cell membranes become waxy. And we know that polar substances like antibiotics find it very difficult to go through a cell membrane if it is high in lipids. In that way, mycobacteria is able to resist most of the antibiotics simply by changing the nature of its cell membrane, thereby reducing the uptake of these drugs into their cells. The fourth biochemical mechanisms by which antibiotics resist microorganisms resist antibiotic resistance with the increased efflux of drugs out of microbial cells. Drugs need to reach a particular concentration and microbial cells to function, which we call the therapeutic level. Without drugs reaching this particular level in cells, they may not be able to function. Now, microorganisms, some of them have developed a mechanism by which they effectively pump out drugs out of their cells almost upon entry. In that way, they reduce the concentration of drugs in their cells below the levels within which they can function. An example is in tetracycline resistance. Most bacteria that resist tetracycline are able to develop pumps in their cell membrane, which are outlets through which these drugs leave almost immediately as they enter, thereby reducing the concentration within the cell and rendering them ineffective. Number 5, biochemical mechanisms by which microbes resist antibiotics would be by bypass of metabolic pathways. Certain drugs, antibiotics like sulfonamides, they target vital pathways in microorganisms, for example, folate synthesis. Microorganisms cannot survive without folate. They can only produce folate. There's no other way they can get it. And so we try to target, to destroy them by targeting their ability to produce folate. Sulfonamide does that effectively, acting as an analog of the substrate power. Microorganisms have been able to resist sulfonamides by simply increasing the substrates which these enzymes may make. And because sulfonamide inhibits the pathway by competing with the substrate, increasing the amount of substrate render the sulfonamide almost ineffective because that inhibition is almost cancelled out. Another way some microorganisms have been able to inhibit sulfonamide is just by changing the enzyme dihydropteroid synthase. This is another target for sulfonamides. So if you are going to inhibit my enzyme, I don't want you to do that, so I just simply change the nature of my enzyme so that the drug becomes ineffective. So by simply changing the form of dihydropteroid synthase enzyme, sulfonamide has been resisted by some microorganisms. The 6 biochemical mechanisms by which microbes resist antibiotics is by overproducing the drug's target. This is similar to the 5th mechanism. How? Somehow, by doing this, they're able to bypass metabolic pathways targeted by antibiotics. For example, intramitoprene resistance, some bacteria are able to overproduce the enzyme which this drug targets, which is dihydrofolate reductase enzyme. So they overproduce in a bid to overcome the inhibition of the drug on this enzyme. So you need to attack my enzyme, I produce more of it, so that the drug binds to some and the others are available to continue to function and produce my metabolite. And then lastly, the 7th mechanism will be the sequestration of the drug. Some bacteria, they just isolate this drug from circulation. They cover it, they hide it, they arrest it, whichever way you want to say it. So some of them can produce methyltransferases that are able to neutralize drugs like aminoglycosides, thereby ensuring that these drugs do not get to their target at all. And without getting their target, they cannot bind. And when they cannot bind, they cannot give us the therapeutic effect we are looking for. I trust that you appreciate these biochemical mechanisms by which microbes are able to continue to outdo us in our search for effective antibiotics to take care of microbial enzymes. What are the consequences of antibiotic resistance? Number one will be limited treatment options. There are so many drugs in the market, but we still have few effective antibiotics. So we continue to struggle with limited options. So many drugs, but only few give us the effect that we are looking for. Another consequence of antibiotic resistance will be a longer period of illnesses. You need to try 1 or 2, 3 or 4 antibiotics before you get the one that actually gives you the relief you're looking for. So that prolongs the period of illnesses from microbes. Another will be higher death rate. That is understandable. Disease that should not kill may kill as we continue to try our hands on different antibiotics to get the desired effect. And then lastly, it leads to increased medical costs. How many antibiotics will you need to try before you get the one that works? So these are some of the obvious consequences of antibiotic resistance we're struggling with. And how can we prevent antibiotic resistance? Number one should be to avoid taking antibiotics without medical prescription. Please do not, when you fall sick, prescribe your own drugs. See a physician. Let these drugs be prescribed before you can take them. Another way to prevent will be to complete your full course of treatment. Should these drugs be prescribed, please do not abandon them. Take the full prescription as prescribed so that these microorganisms do not survive at all in you to begin to have the opportunity to develop resistance, waiting for further future attack. And that will be to improve hygiene and infection control measures. And then the government should promote the use of vaccines against microbial infections, and also should invest in future development of new drugs. We need to invest in the search for new antibiotics that will work. Thank you so much for your time today as we have learned about antibiotic resistance. I'll see you next time. Thank you. Bye-bye for now.